Okay, so good afternoon everyone. Welcome to Trieste. I actually work at the ICGEB since many years with Professor Baralle, and we've always been an RNA lab, and for many years we have been working on the impact of RNA splicing mutations in disease. So thanks to the excellent introduction this morning by John, I don't really have to say anything more about the splicing process. You already know in detail what it is. What is important to understand, I think from a human health perspective, is that the splicing process normally is engineered to actually give a, where is the point? The pointer, normally is engineered to be extremely precise, actually at the nucleotide level, so that if you have a pre-mRNA that has to code for a protein, all the exons that specify for the translation of this protein, and especially they have to be joined together so that the translational code is preserved. And so all the great complexity of the spliceosome is really there to make sure that this open reading frame is carried forward without any mistakes to the ribosome and the protein being produced. Now, when this doesn't happen, as you can well imagine, there starts to be problems, right? Especially at the level of disease. And normally, this is due to the fact that in a random manner, mutations can actually affect all the specific consensus splice sites that tell the spliceosome where an exon is beginning and where an exon is ending. And you can see here that at a very basic level, of course, we have the 3' splice site, the branch site, and the 5' splice site, and these are recognized by some basic factors who actually tell the spliceosome that the exon is beginning at this nucleotide here and is ending exactly at these two nucleotides here. Then very often what has been found is that these basic splice sites are not sufficient to give the spliceosome enough information to recognize the exon as a unit that has to be kept, and therefore there is a number of regulatory elements that can be in the exon itself, but also in the neighboring intronic sequences, and these sequences are called splicing enhancers or splicing silencers. And they bind those proteins that Silvano Riva just talked to you about, these SAR proteins and HNRMP proteins. So if by chance you have a mutation that alters the consensus site for the binding of these basic elements or these other more advanced elements, then the exon is not recognized anymore. And when the exon is not recognized anymore, what you have is a series of problems with the processing of the pre-mRNA. And of course, the most common and the most common defect that you find is that if you have a mutation inside the consensus sequences, then the exon is not recognized anymore. So what we call this event is exon skipping, and very often when one exon is skipped, you can either have two outcomes. Either you have the modification of the open reading frame, so the protein is not translated or is translated in a truncated manner, or you can also have maybe if the exon is in frame with the upstream and the downstream exons, what can happen is that then your protein will be translated, but all the sequence information that was present in the exon will be missed. And so probably very often the protein becomes non-functional or it acquires functions that it shouldn't have. Then the other most common outcome when you have a mutation that is connected with disease is the fact that only one of the splice sites is impaired, the other one is kept in place. And when this occurs, what the spliceosome does, it tries desperately to find some sequence that looks like the original splice site that has been inactivated. And so the result is that you do have the inclusion of the exon, but this exon will either be shorter than the original or it will be longer because it will include a little bit of the flanking intronic sequences. And again, what you can have at the level of protein is a change in the open reading frame, so very often the production of a truncated protein, or the addition or deletion of some of the residues, again with the possibility of inactivating the functions. And then there is another class of splicing mutations that really was really underappreciated until some time ago, when the power of total RNA sequencing wasn't still available to researchers. And this is that a lot of mutations, when they fall into very long introns, what they can give rise is that they can actually create false splice sites. And so inside very long introns, in many cases of disease, what you do find is that you have the insertion of extra exons that normally wouldn't exist inside the mature mRNA, and therefore you get a protein with that where the open reading frame very often is changed, or where it has extra amino acids, stop codons, or so on, that disrupt its function. So how often do splicing mutations of this kind occur? And this is a review that I wrote a few years ago, and basically the bottom conclusion that you can get from looking at many kind of diseases and look at when you have mutations, how many of these mutations can affect the splicing process, you get an average of 10 to 15%, but it can vary from as low as 3 to as high as it has been seen 50. And of course, this wide range of percentages is due to the fact that in exons where you have many, in genes where you have many exons and you have very long introns, it is more likely that you will find splicing mutations. But for genes that are smaller and have shorter exons, then splicing mutations are not that common. But the bottom line is that any gene in our body that is subject to splicing can actually be also subject to splicing mutations, and these splicing mutations will eventually give rise to disease. So one of the things that we have been doing in the lab in the last few years, and we have done this quite extensively, is to actually look for splicing mutations. And how do you look for splicing mutations? Well, the easiest thing that you can do is you can simply look at the gene of interest. So if you have a patient that is suffering, let's say, from neurofibromatosis, what you can do is you can extract blood cells, white blood cells from the patient, and you can amplify the mRNA of the patient, and you can look whether some exons are skipped, if there are extra exons that are included, and this way you can understand, right, why the cause of the disease. But of course, this is not very easy to do. First of all, because many times you cannot get hold of the patient, right? The patient may be living very far away, and therefore it is quite difficult to get an mRNA sample that you can amplify directly using RT-PCR to actually check for the splicing mutation. The other reason, of course, is that if you suspect that the splicing mutation is actually affecting a particular gene, then really you have to make sure, if you want to use this technique, you have the prerequisite essential requirement is that this gene should be expressed in blood cells. If you have a gene that is only expressed in the brain or in the heart, it is actually very, very difficult to get an RNA sample to analyze directly. And for this reason, especially in the lab where I've been working in since many years, what we have done is to set up an artificial system, and these artificial systems now are very common. They are used to analyze splicing mutations all over the world, and they're called the mini-gene system. So what happens in the mini-gene system, you try to actually mimic the splicing process of the gene of interest in an artificial manner. And you can do this because if, for example, you have, again, a patient that you think is affected by neurofibromatosis, so you sequence the DNA of this patient, you find that there might be a splicing mutation affecting the recognition of the exon, let's say 13. And so what you do, you get your exon 13, you get some of the flanking intronic sequences, and you put it into this artificial construct where you actually have your exon of interest, the flanking intronic sequences, and then you have other exons that are constitutively recognized, and you can form in this way a small pre-mRNA that you can, in a cytoplasmid, that you can then transfect into cells. You can allow the pre-mRNA to be transcribed and be spliced by the cellular machinery. And of course, you do normally you do two mini genes, one that carries the mutation, the other one that doesn't, and then you see if there is a difference, if the mutation actually makes a difference in the recognition of this exon. The other thing that has been done from all the literature on splicing mutation is to actually, and I'm telling you this because this is something that is quite common nowadays also when analyzing splicing mutations, is to actually use bioinformatics. So one of the ways that people now have started analyzing all the many different variants that could be splicing mutations and could be connected with disease is to actually develop some bioinformatic tools where you input your sequence of the exon, of the flanking introns, and these bioinformatics tools will automatically check for you whether that particular mutation is likely to affect either the 5' splice site, the 3' splice site, or some enhancer sequences. And in this way, without doing any experiment, you can actually start to, if you have a long list of mutations that you want to analyze, you can start to use the bioinformatic to prioritize the mutations that you want to analyze using more conventional means like the mini gene or directly from the patient. And this year, what I wanted to do to all of you is to change a little bit the format of my talk, because since many years we have been looking at a particular metabolic disease that is called Pompe disease, and our research on this disease actually recapitulates completely all the approaches that can be used when someone wants to investigate splicing mutations. So from now on, I will be talking specifically about Pompe disease, but please keep in mind that the approach that I will show you in the next 20 minutes or so can actually be applied to any gene of interest. So what is Pompe disease? Pompe disease is an autosomal recessive lysosomal storage disease. It is quite common. One person, there is one, the frequency is 1 in 40,000. And basically, you develop Pompe disease when you have a deficiency in a particular enzyme that's called alpha-glucosidase. And what alpha-glucosidase does is something very simple, and what it does, it mediates glycogen degradation into cells. So when this enzyme is not working anymore, right, what you have an accumulation of glycogen within the lysosome of the cell, and this one, if it's carries on and becomes more and more severe, can eventually give rise to cardiac and skeletal muscle problems. There are two forms of this disease. One is the infantile onset, and normally these are very severe form of the disease, so that the poor children who are born with the infantile onset die very soon after birth. And then there is a late onset that occurs in people who are much older, even when they are in their 40s, in their 50s or 60s. And the late onset actually shares some of the problems of the severe onset, but there is no cardiac involvement, there is some problem with the skeletal muscles, and eventually people, if they die of respiratory insufficiency. On the other hand, the infantile onset is characterized by very severe muscle weakness, a very enhanced cardiomegaly and cardiomyopathy, and respiratory insufficiency right from the beginning, right? So why is this a nice disease to actually investigate? And I'll come back later on this subject, because what I'm going to do is show you how, after you have studied the splicing mutation, of course the big question is, what do we do about it? How can we develop a therapy based on correcting this splicing mutation? And the reason why this is a very interesting disease as far as to study is that you have a lot of room for improvement, right? You start having the disease only when the levels of acid maltase actually drop below 30% the normal level. And even then, there are a lot of people who have less than 30% enzymatic activity with respect to the healthy controls, but are still, quite healthy. So it's only when you go below 30%, and of course when you carry some other, let's say, risk factors, that you start developing the disease. And what this means is that if you can, from a therapeutic point of view, right, this is very advantageous, because what this means is that if you can improve just a little bit the amount of enzyme that is produced, you can quite likely change a adult onset into a perfectly healthy individual, or an infantile onset to an adult onset that is much more easy to manage, right? So at the moment, just let me tell you, before 2006, there was nothing medical people could do if you were diagnosed with Pompe disease. After 2060, there is an enzyme replacement therapy. So what actually the only therapy that is available, and that is quite effective, is to actually make the enzyme artificially into CHO cells and then give it to the diseased people, right? This is quite, of course, it's very expensive, right? So this is certainly one limitation. And the other limitation is that it is effective, but not completely effective. So there are ways, so people have been looking for alternative ways of therapies. Now, if you look at the gene and you look at mutations, what has been found by many epidemiological studies is that you have over 200 mutations that actually affect these genes. Now, many of these mutations are found only in very few individuals, and many of these mutations, of course, affect not just the splicing, but also the amino acid coding capacity of the gene, so I won't be talking about this. But one of the very interesting findings from these epidemiological studies is that in about 40 to 70% of all the people who have late onset of the disease, well, what these people have is they have one allele that is completely inactivated by a very severe mutation, but the other allele carries a splicing mutation. And this splicing mutation is written here, and I'll show it to you in a second, but basically it's a single nucleotide substitution in the 3' splice site, where at nucleotide 13, before the acceptor site of exon 2, there is normally a T, and this T has been replaced by a G. So all the people who carry this mutation actually keep a little bit of the ability to make the enzyme, and this small ability to make the enzyme actually turns what could be a very severe disease into a late onset one. We started working in 2006 actually on Pompe disease, and we started working like you normally do on when you start a project on the connection between splicing and disease. You go through the register of all the people who have been described to have Pompe disease in Italy, and you look at the mutations, right, and you see which ones could be splicing mutations, which are probably missense mutations, which are truncation mutations. And by doing this screening, what we did find was a number of new splicing mutations that we then characterized with the minigene. So these changes, we thought they were splicing mutations, but of course we had not access to the mRNA of the patient, and because some of them had actually died some years ago, we only had access to the DNA, and the bioinformatics was not actually giving a very good yes or no answer. So the bioinformatics was saying, well, they could be splicing mutations or maybe not. So what we did, you can see here, we took these 3 mutations that we didn't know if they could affect splicing or not, and we made 3 small mini-genes, right? There was one mutation in exon 7, the other in exon 11, the other in exon 18. So we made 3 small mini-genes that were made by keeping exon 6, the intron between exon 6 and exon 7, the downstream intron, and then exon 8. And using this mini-gene, we transfected it into a series of cell lines, like HeLa, COS1, CHO, Hep3b. And then we looked whether in the presence of the substitution that could be a splicing mutation, there was a change in the recognition of the exon. And as you can see here, for all 3 putative splicing mutations, we had a positive answer, because whenever you have the wild-type sequence, exon 6, 7, and 8 are perfectly joined together. You can see it from the band that is up here, and that is composed by these 3 exons perfectly joined together. But whenever we put inside the putative splicing mutation, then exon 7 was completely ignored by the spliceosome, and only 6 and 8 were joined together, so giving a defect. Then, 2 years ago, what we started to do is to actually stop doing this work because it was getting a little bit boring, and we decided to concentrate on that -13 mutation, because the -13 mutation was the one that gave us the greatest possibility of actually starting to think about a therapeutic approach. And so what we did was to, first of all, there was very little known about this mutation. The people who first described it in 1994 had done a preliminary characterization, so they saw in their manuscript that when you had this G replacing a T here, what happened is just what I told you. There were several missplicing events that were taking place. So there was the skipping of the entire exon 2 that is very long. It's uncommonly long for an exon. And also there was because this 3' splice site was inactivated, associated with the skipping of the entire exon, the splicing machinery was also recognizing an acceptor splice site that is here, 60 nucleotides before the donor splice site, and that in normal patients doesn't seem to be, is not recognized over much, but in patients where this 3' splice site has been impaired, then it becomes one of the major splicing outcomes. And we confirmed this by looking at the splicing profiles inside 2 patients that were only expressing the allele carrying this mutation with respect to controls. And you can see here that in the 2 patients, basically the only 2 splicing species that could be detected were the ones where the exon 2 was totally skipped, and the one where only these 60 nucleotides were retained. But in the controls, you did have a little bit of skipping, showing that this is a very difficult exon for the spliceosome to recognize, even in the best of conditions. But you do also have a quite significant amount of normal splicing going on, so the entire exon 2 being recognized. Of course, we were interested in knowing at the molecular level what this mutation could have affected, and that was not very difficult, because as this mutation was localized only 13 nucleotides before the acceptor splice site, it is well known that during the early stages of exon recognition and definition, the 2 factors that are required to have a good recognition of the acceptor splice site are shown here, and they are U2AF35 that recognize the AG, and then U2AF65 that recognize the polypyrimidine tract that is just upstream of the AG. And so what we did was to test using 2 small artificial RNAs whether this replacement of a T for a G was actually affecting the binding of the most likely culprit, that is U2AF65, and you can see that the answer was immediately positive. So this is the RNA that carries the natural U in the -13 position, and you can see that U2AF65 can be combined quite well to this mRNA without any problem. But if you use exactly the same RNA oligo replaced where this U has been replaced to a G, then the recognition of U2AF65 is completely lost. So what we thought of doing actually is to try to understand what were the auxiliary factors that were responsible for the recognition of this exon. And of course, I will show you later how we plan to do this in a much more comprehensive manner. But the thing that we did, to tie my talk with that of Silvano, is to actually try the most common splicing factors that can be found inside a cell. So several SR proteins that normally promote exon recognition, and several HNRMP factors that sometimes act as negative regulators and sometimes also as positive regulators. Now, this was very difficult to do in the patient cells, so what we had to do is to actually make a mini-gene, right? And what we did, the mini-gene, you can see the basic scheme here, so we forgot about the rest of the GAA pre-mRNA. We simply got the exon 2, we put it in inside our mini-gene, and we, of course, made two kinds of mini-gene. One that was carrying the U, so the normal mini-gene, wild-type sequence, and another one that was carrying the G, the mutated sequence. And you can see that immediately when we transfected this mini-gene, we got exactly what we expected, because in the case of the wild-type sequence, we actually could see some normal splicing of the entire exon being included in the final mRNA, but with the mutated, then we couldn't see anything using a straight RT-PCR approach. If we used a quantitative approach, we could see that there was still some residual recognition of the entire exon 2 that was around 20% with respect to the wild type. So we took these two mini-genes and we co-transfected these two mini-genes with a whole series of SAR proteins and HNRMP proteins, and we were simply interested in knowing which of these factors was capable of improving in the mutated mini-gene the recognition of the whole exon 2. And you can see from these panels here, these are RT-PCR experiments, you can see that only one factor, called SRP75 with the old nomenclature, but SRSF4, was actually capable of improving recognition of the normal exon 2 in the mutated context. And we, of course, then wanted to check whether this could be actually carried forward in the patient fibroblast that we analyzed at the beginning. And you can see that indeed this was actually true also from the patient fibroblast, because when we overexpressed this factor, SRP75, called SRSF4 now, in the patient fibroblast, there was an improvement in the amount of enzyme of exon 2 that was being processed. And also, if we did the reverse approach, so we knocked down SRSF4 in the patient fibroblast, then the residual amount of normal exon 2 that was included in the mRNA was actually decreased in a significant manner. So what we did, now that we know a name, what we could do is we went through the literature and we looked for all the compounds that could actually affect the levels of SAR proteins, and especially SR75, right, in an artificial manner. There are several synthetic and semi-synthetic compounds that when you give to cells will have an effect on the levels of SAR proteins and HNRMP proteins that are expressed by the cell itself. And so we went through the literature, we selected the ones that were most likely, and we tested them, first of all with the mini-gene, and then also in the patient fibroblast. And what we found is that out of all the compounds that we tested, one that is called resveratrol was quite efficient, you see, in stimulating the recognition of exon 2. This one was in the minigene setting. So what we did was actually to immediately go to the patient fibroblast and treat this fibroblast with resveratrol. And you can see that these are the two patients that were tested initially. When you treat the patient fibroblast with resveratrol, if before you had very, very tiny amounts of normal GAA enzyme being produced, following the addition of resveratrol, you can see that you start having an improvement in this level of production, and this production was actually viable, because if you tested the patient fibroblast after resveratrol treatment for an enzymatic assay that you can see here, you can see that in both cases we could actually achieve an improvement in the production of an improvement in the enzymatic activity. So we then carried on testing for other compounds. We found a few, but obviously, right, what we want to do now that we have, let's say, established this kind of proof of principle that we can actually use artificial compounds to improve the production of this enzyme in the patient fibroblast, what we are trying to do now is to actually use this approach to screen for in a much wider manner. And what we have done is actually engineer, and this is the work of basically the last 3 weeks ago or so in our lab, is to actually engineer a different kind of minigene that can be suitable for such a purpose. And this is something that now is commonly used for many splicing mutations, interesting splicing mutations. And basically, what we have done is to actually take a plasmid that was expressing a green fluorescent protein, right? And in the middle of this green fluorescent protein, we cloned the exon 2 of the GAA gene with a little bit of flanking intronic sequences. So we had to make artificial 5' splice site and 3' splice site here at either end. And this actually new minigene was constructed in such a way that if the exon 2 is skipped, then the two halves of EGFP are joined together and can give a fluorescent signal. But in the wild type condition where most of exon 2 is included, then exon 2 will be retained in the final process, the mRNA, and therefore the green fluorescent protein will not work anymore. And we have tested it as a proof of principle inside by transfecting just heal a cell line, and you can see the result here. Basically, if you transfect this kind of minigene with the wild type normal U in the position -13, you don't have any production of EGFP, but if you transfect the minigene that carries the G, so the mutant allele, then you start to see EGFP production. And you can see, also visualize this very, very efficiently by immunofluorescence. And also, what we tested, because we already knew that resveratrol was effective in the other minigene context, we tried to transfect the mutant minigene in the presence of resveratrol, and what we expected is that if there was an increase in exon 2 inclusion, then the level of EGFP being produced would be reduced, and this is exactly what we saw. So what we are going to do now is to actually make stable cell lines with these fluorescent minigenes, and these stable cell lines we will select using a high throughput approach for thousands of different compounds or thousands of different factors to actually find the ones that are more efficient in restoring exon 2 inclusion in the presence of the mutation. And in parallel, what we are trying to do, because testing in patient fibroblast is fine, but of course, if you really want to look for a viable therapy, what you want to do is to get as close to the patient as possible. And so what we are setting up is an approach whereby we actually, from the skeletal muscle cells biopsies taken by the bank of patients that is available in Italy, what we're trying to do is to actually extract multipotent adult stem cells that can be differentiated into myotubes. And you can see here that this is something that we have already managed to do, because if you have the normal fibroblast, they don't, of course, produce any of the specific muscle proteins, but if you have these multipotent adult stem cells isolated from the patients, you can grow them into myogenic medium, right? And if you do that, after 28 days, you can see that they start to express myosin, where they didn't, of course, at the beginning. And also, if you look at the cells under the microscope, you see that they start to have the appearance of multinucleated cells. That is just what happens in normal muscle. So after we have done the screening and we have found some very suitable compounds, what we plan to do is to actually test these compounds into this very advanced system, and eventually, of course, then go on to the patient. So I think I'll stop here, again, reminding you that this kind of approach, you just have to change the name of the gene, of the exon, and of the mutation. You can apply it to almost any splicing mutation you can think of. And just thanking the people who are responsible for this. Of course, Tito Baralle, who, nothing of this one would have been done without him, and Cristiana Suani, who is our technician, and Najed, that is not here because she comes from the BDG, who helped us with the myotubes. And with this, I thank you. And